This is the video lecture for um, the class that would have been on Thursday, March 19th. There will be two video lectures for microbiology for biology majors. And today we're going to start on chapter 24. I hope you're all doing well. This is really an odd time. Even though we're supposed to shelter in place, faculty and other university professors are able to teach. So anyhow, we'll just keep the materials coming and I'll keep you informed on deadlines and so forth. So anyhow, the objectives, this is chapter 24 and it is up in your lecture notes. So the objectives here are to talk about ecology. I know you've all had ecology or pretty sure you've all had ecology. So this will be a review for you. We'll talk about different bioelements and how they're cycled and how microbiology plays a role. And we'll talk about microbes on land, water. There are microbes in air. They're a little more rare and a little bit more difficult to characterize, but we're going to limit our discussions to water and land. Okay. And so when you talk about microbial ecology, this is the study of microbes in natural habitat. So you're looking at microbial communities that are mixed communities. And in their habitats, they have biotic factors, but these biotic factors are living or dead organisms habitat. Okay, and then the non-living components are abiotic factors. Okay, and these abiotic factors, this would be like the difference between a community and an ecosystem. So an ecosystem includes non-living and living, whereas a community only includes living. Okay, and so roughly we can divide the Earth up into three spheres: the lithosphere, which is land, the hydrosphere, which is water, and then atmosphere, which is air. And all of these comprise where you would find the biosphere, where you would actually find life. Okay. And if we start at the top and work our way down in terms of the ecological hierarchy, then you start up with the biosphere. And then if you look at specific zones, these are called biomes. And so, there are different zones that are based on different latitudes, northern and southern latitudes, where as you get closer to the equator, you would have things like tropical forests, and then you would have deciduous forests, and then taiga and tundra as you're going further north. And then if we look at an individual section of a biome, we would call this an ecosystem. Okay, ecosystems have probably more arbitrary boundaries associated with them. And then within an ecosystem, then you have a community, and a community includes only the living organisms. And then finally, if you look at a single species, like Chlamydomonas, then that single species, that's a protozoan that you find in freshwater, and that would be considered a population. A population is only of a single species. It's like if we count the population of redding, we're only talking about humans. We don't talk about humans and dogs. So we don't cross species when we talk about a population. And then finally, the individual organism. Okay, so biomes are laid out in terrestrial and aquatic realms based roughly on latitude. Okay, and also altitude and dominant plant form. You can have tundra and taiga at latitudes that are closer to the equator, but they would be at high altitude. Okay, so that would include something like a grassland. a desert, a mountain, okay? And biomes are essentially generally balanced, okay? So they're in some type of equilibrium, okay? And each organism has a habitat and a niche, okay? Its habitat is where it lives. Its niche is its role within the community, okay? And in natural environments, you only see mixed cultures. It's very rare that you would see a pure culture in the laboratory. And so some of the things that we do in the laboratory are um, really artificial because we see single species of organisms. But in natural environments, including the gut biome, you would see a mixed culture. This is an interesting bacterium, uh, the Sulfurudis odd axiator is breeds gold. It's a gold-reducing organism. Okay, it does exist as a single species. I just thought this was kind of cool, so I included it. Okay, and so if you look at another way that we can capture the hierarchy of different organisms, we can talk about producers and consumers. Okay, so producers are autotrophs, consumers are heterotrophs. Okay, and consumers are also, we have a subgroup of consumers that I would consider a subgroup called the decomposers. And then after the primary consumers, secondary consumers, tertiary consumers, and quaternary consumers have consumed materials from the producers, then material starts to die and decay. So these would include saprobes that are decomposing dead and decayed material. And as you go up this hierarchy, then the amount of available energy becomes less, and the populations actually become lower. You have less and less quaternary consumers as tertiary, as secondary, as primary producers. Okay, and so you can see a diatom would be sort of a primary producer. Paramecium would consume the diatom. Didinium would be a secondary consumer, would consume the paramecium. Cyclops would consume the dididium, and an insect larva would consume the cyclops, and then the minnow would be at the top, would be the top carnivore, and would be at the top of that particular food chain. We know that's not the very top because other fish are going to prey on the minnow. Okay, really, the food chain is more exactly um, described as a food web because there's a lot of different interactions in the community. There's a lot of different predator-prey relationships. And so rather than describing this as a food chain, probably when you had ecology, then this was more described as a food web. And all elements recycle. So you have natural recycling of bioelements. So remember, mass is conserved, so there's only a finite amount of carbon, hydrogen, oxygen, and so forth on the Earth. So when things are moving and changing, they're moving from organism to organism, they're moving from living to non-living, primarily in the lithosphere and the hydrosphere. Okay, so elements go back and forth between abiotic and biotic. So an element can be tied up in a living organism or it can be tied up in something that's non-living. Okay, and these cycles are interdependent with the food web because the food web then is providing materials that will eventually become dead and decaying materials, so will go back into non-living. Okay, so all organisms participate directly in bioelement recycling. Even humans, we're recycling bioelements all the time. We're recycling oxygen and carbon dioxide, obviously through respiration. we're eating and excreting, and so there's sort of this biotic abiotic recycling that we're doing at all times. Okay, and only some organisms can work with something that's completely inorganic. Humans generally can't. There are a few things that we can cycle directly, but there are just a few organisms that can go from an inorganic compound and move it into something that would be nutritional. Okay, so here is how carbon is sequestered and how carbon makes it into the atmosphere. Okay, so we have lots of processes, and this is obviously a real hot button issue right now because of the human or iatrogenic carbon release into the atmosphere, including combustion and respiration. Trees actually respire, decomposers, consumers, producers, everything respires. The volcanic geothermal activity will also release carbon dioxide into the atmosphere, and in some cases can comprise the majority of the carbon dioxide in the atmosphere. And then the sink is the producers of the photosynthetic organisms, and they'll take carbon dioxide out of the atmosphere. So it will cycle directly up to the atmosphere through state probes and through consumers and through abiotic sources like volcanoes and combustion, and then it will be taken out of the atmosphere through producers, including primarily photosynthetic organisms. So in the carbon cycle, oops, the principal users of CO2 are photoautotrophs. Remember, photo and chemo refer to energy, auto and hetero refer to carbon source. So this is an organism that gets energy from light and gets its carbon from inorganic sources, from CO2. This accomplishes photosynthesis, which builds up organic compounds. And then finally, you have respiration working in opposite, along with fermentation that breaks down the organic compound. Okay, so that goes in the opposite direction. And then there are a few secondary roles. There's a lot of limestone, especially in the west, in the soil, and that liberates carbon dioxide in the water, and that goes into the atmosphere. And then methanogens can liberate a large amount of methane. Okay. As far as the nitrogen cycle is concerned, it's pretty complex here. So basically, you start out with nitrogen gas. Nitrogen gas can be fixed by nitrogen fixers. Okay, these include primarily bacteria in the root nodules of plants. Okay, and other types of bacteria. These will produce ammonia. Okay, and then ammonia can be nitrified to produce nitrite and then nitrate. Nitrate is a wonderful fertilizer for plants, algae, and denitrifying bacteria. And so then that will be converted to organic nitrogen through things like amino acids and nucleic acids. Then the organic nitrogen then can be consumed by animals and protozoa, the consumers. As they give off waste and decay, that will result in ammonification, so they'll get more ammonia. And then nitrifying bacteria can also cycle between organic nitrogen and ammonia. Okay, so you can see it's very, very complex. And then all of this in terms of degradation, things like denitrifying bacteria then can take nitrate and nitrite and produce atmospheric gases, nitrogen gas. So that completes the cycle at step 6. Okay, so in the nitrogen cycle, it's more like a web. It's pretty complex. So organic products, waste products, including urine, and as well as that in decaying material produces ammonia. Nitrification takes ammonia and it oxidizes it to nitrite, and then following with that with nitrate. Then nitrates, which is a great fertilizer, can be used by plants and bacteria and other organisms. And then denitrification then will take it finally to nitrogen gas, which is released in the atmosphere. As far as sulfur is concerned, sulfur can exist as elemental sulfur in a rock, hydrogen sulfide gas, sulfate ion, and let's see, what's the best way? Disulfur trioxide. Okay, and plants can only assimilate the SO4 ion, and this should be 2 minus if we're technically correct. It's not, this is an ion, so, and it doesn't have an ionic formula, but it is 2 minus. Animals can only assimilate organic sulfur, so it has to be converted to an organic form. Thiobacillus will take sulfur directly from things like iron pyrite and make sulfuric acid from an intermediate called thiosulfate. And this is what happens up in Iron Mountain Mine. Iron Mountain Mine is a Superfund site. It's a really, really toxic waste site about 9 miles out of Redding. It has the lowest recorded natural pH in the world. And it's a real problem because the bacteria can produce sulfuric acid directly from what's going on in mine drainage. Plants can use sulfuric acid, that sounds weird, in very dilute forms. Usually this is more of a sulfate salt than sulfuric acid, but plants can convert the sulfate directly to organic forms. And then bacteria called Desulfovibrio and Desulfuromonas can take sulfate and like SO4 and convert them to hydrogen sulfide gas and sulfur. Hydrogen sulfide gas. This was one of the experiments that didn't work. Proteus vulgaris produces hydrogen sulfide gas from sulfur, and it's one of the few bacteria that we use that actually can do that. That was a on iron deep that was supposed to turn black, and it didn't. But you did see the demonstration. That's just a property of Proteus vulgaris and slightly for Pseudomonas aeruginosa. In terms of phosphorus cycling, the chief inorganic reservoir on Earth in the lithosphere is fluorapatite, and fluorapatite has a phosphate group in it, PO4. Okay, and this chemically releases PO4 ion, phosphate ion, which should be 3 minus, by acid erosion of fluorapatite. Thiobacillus also dissolves phosphate rock and will release phosphate into liquid phase. And then organic phosphate, which is really important for cell signaling. It's also important for membrane integrity, phospholipid bilayers. Is then organic phosphate then is broken down to PO4 3 minus by decomposing organisms. Okay, enough with cycling. Let's talk about soil microbiology. So the soil has quite a pore space. The porosity of soil is almost 50%, and even higher in things like sand. So at the surface of the soil, because soil is permeable and has lots of pore spaces, then you would find lots of oxygen. Okay, especially in dry soil where it's not saturated with water, where the oxygen can permeate a little bit easier. Dry soils are really great for oxygen, but as you increase in depth of the soil, as you get below the soil surface, then you have less oxygen and you have more types of anaerobic metabolism. And carbon dioxide, which is being released from carbonate and calcium carbonate in the soil, will increase with depth of the soil. And so as oxygen is depleted, then the next thing that you see in the soil is denitrification, followed by sulfate reduction, followed by iron and manganese reduction, and then finally methanogenesis, where the electron acceptor is actually carbon dioxide. So in each one of these, you actually see zones of different bacteria that will metabolize first oxygen and then breathe nitrate, then breathe sulfate, then breathe iron 3 or manganese 4, and then breathe carbon dioxide. So if we see the porosity, the pore spaces, you can see a lot of activity in the soil. You can see dust mites, clay and silt. You can see protozoa. You can see nematodes, which are a type of helminth, and then you can see bacteria. And there's a lot of organic carbon in the subsurface, and so the bacteria are going to gather on the organic carbon. In terms of microbial activity, we look at the rhizosphere, which is just the zone of soil surrounding the roots of plants. Okay, there's a lot of activity. It's generally aerobic. The roots of plants are really good in terms of creating pore spaces in the soil to allow oxygen to flow in. And there's a lot of different symbiotic relationships, say between bacteria and fungi, releasing growth factors for plants, and then a lot of nitrogen and phosphorus cycling in terms of nitrifying bacteria, acid production from fluorapatite, where phosphoric acid then can be released for the plants to use. Okay. Fungus will actually extend the root zone of plants, and you get what's called mycorrhiza. They attach themselves to roots of plants. These basidiomydes, ascomides, and zygamides are all different types of fungus that will actually attach to the roots of the plant, and they can scavenge for water and provide water to the roots. And the plants feed the fungus via photosynthesis. And then the mycelium or the fungus will extend beyond the plant, and that helps the plant capture water and minerals. Here's mycelia. You can see the plant roots here that are more dark brown. Mycelia is white. And so that it just extends the water capacity here. Again, so you have even further roots of mycelia that are acting to transport water and nutrients to the roots. In terms of topsoil, you'll find growing animals, so you get nematodes, termites, and earthworms, or more macrobiology, decomposing and reducing organisms. And then because you're at the top of the soil, then you would expect more aerobic processes. And so you see aerobic microbes that will finish the whole process of decomposition and get to the level where you're producing minerals or pure compounds that cannot be reduced any further. Also, there's a lot of fungal activity, like enzymes that fungus that produce enzymes that will break down things like cellulose in trees, keratin in animals, lignin in plants, chitin in paraffin, not paraffin. And here's some nematodes. These are small helminths, okay, and you would find these in the topsoil. And then as you get into the deeper subsurface, then when the oxygen becomes limited, then you have primarily anaerobic activity, and they've adapted to different terminal electron acceptors. And the first zone, below oxygen utilization, then you would see denitrifiers, which can use nitrate and nitrite. Then next you would have manganese reducers and iron reducers, then sulfate reducers, then methanogens. And if you look at a zone, this is a zone of contamination, and you get further and further away from the carbon source, the organic carbon, then you'll see that there are actually zones of microbes out here far away from the contamination. It's still aerobic. And then as the contaminant seeps out, then the oxygen is used, and then it will go to denitrifying here, then manganese reduction here, iron reduction, sulfate reduction, and then finally, after all of those electronic stores are depleted, then and only then you'll get methane. So you may wonder, what does the atom bomb have to do with microbiology? That's a very, very interesting question. And I used to work at a place called the Hanford Nuclear Reservation, and that is where they made the plutonium for the atomic bomb that was dropped on Nagasaki, not on Hiroshima, but on Nagasaki. And the whole process of making plutonium, here's an old picture of the Hanford Nuclear Reservation in eastern Washington. And within the whole process of what was called the end of the World War II and the Cold War, the country was focused on making armaments, and the really focused on the arms race with the Soviet Union at the time, and not focused on environmental cleanup. So a lot of times what would happen is the waste that was associated with making plutonium would just get thrown into the dirt. So you would see boxes of different types of materials and tanks or barrels that might have special nuclear materials in them, and they were thrown in what were called unlined cribs. And the unlined cribs were just basically holes in the dirt. Okay, so this created an environmental catastrophe. Here is what's called the 300 area at the Hanford Nuclear Reservation. And at that, no, that's really the 200 area. And in the 200 area, what happened was a massive, massive problem with contamination with nuclear radiation, special nuclear materials, and also waste like carbon tetrachloride and sodium nitrate. And so if you were at the nuclear reservation, because of the contamination, then you would suit up with something like this. Ultimately, you would be wearing a hat or a hood at the same with this outfit, and you can see that with this individual, their boots are taped up directly to their hazmat suit, and just to prevent anything leaking in and prevent any type of direct exposure with the air. Okay, and this is where the nuclear reservation was, and its proximity to the Columbia River. And so what happens is when contamination goes into the groundwater, goes through the soil surface all the way down to the groundwater, then ultimately the contamination will flow towards the Columbia River. Groundwater is just an underground river, and the underground river flows to the surface. And so the big problem at the Hanford Nuclear Reservation especially was the fact that carbon tetrachloride was very, very heavy. It leaked into the groundwater, and then it started, the goal was to prevent it leaking into the Columbia River, which was the water source for about 250,000 people. Okay. At the Hanford Nuclear Reservation, when they had special nuclear materials, they would store them in these very, very leaky, unlined tanks. And these tanks were insufficient. You can see that they're rusted out. In fact, all the materials had to be pumped out and pumped into double-walled tanks, which are still there to this day. And there's a whole issue with the nuclear materials and some of the acids, as well as some of the solvents solvents outgassing, and actually the entire tank would burp, which would cause a small tremor, or earthquake when the tank burped. And so I was involved in the design of the new tank farms and helping tackle the problem of the tanks burping. Okay, so how do contaminants get there in the first place? So what you would do is there would be an underground tank. Okay, the underground tank would be leaky, so the contaminant would leak out. Okay, they also had an unlined crib, which was just a hole in the dirt where they would put the different materials in. That would leak through the different layers of the soil, and then ultimately get below the water table here and get into the water. Okay, and what could be done in order to prevent contamination from going directly to the Columbia River was to drill wells and monitor the progress and make sure that this contamination wasn't flowing fast enough that it would be a problem for the Columbia River. So this had to be remediated. Different types of remediation include bioremediation, and this is where microorganisms that are naturally in the soil already will both in situ and ex situ. In situ means on site, ex situ means that you actually take the contaminated dirt or soil or water and you remove it and you treat it at some type of waste treatment facility. So in situ, treating the contaminants in place. Ex situ, excavating the contaminated site and treating the soils and water at a separate location. Okay, and then in some instances you can use plants. The roots of plants are really, really good at decontaminating surface soils. Of course, the roots don't go down very, very far, so if you have really deep soil contamination, then that would be a problem for phytoremediation, and you would revert to bioremediation and just use microbes. Okay, so if you have native organisms in the soil already, then typically they've been naturally selected over time in the contaminated zone to both survive in the presence of the contaminant and in some cases eat the contaminant. So all you have to do is find the right nutrient mix to stimulate the native organisms to also eat the contaminant. Bioaugmentation is a little different. When you have contamination where you don't have microbial activity that can remediate the soil, you may have to directly inject microorganisms into soil and water. So stimulation, the microbes are already there. Bioaugmentation, you have to add them directly to the contaminated site. Okay, bioventing, sometimes just by taking a zone of soil that's anaerobic and injecting oxygen to the soil and the groundwater, that will stimulate aerobic organisms. And this is really good for petroleum hydrocarbons. A lot of times when they have old gas stations that had leaky tanks where gasoline leaked out, all they will do is they'll drill holes into the contaminated area and they'll just pump air down there, and that just the action of pumping air will stimulate aerobic organisms to eat the gasoline. Also, land farming. This is when contaminated soils are just tilled periodically, and they're tilled basically to bring soil to the surface so it can be aerated. Also, composting. This is just piling up the contaminated soil. Sometimes you mix it with other things like leaves and litter, and that helps to promote aeration as well. Okay. And when we specifically talk about biostimulation, this is stimulation of native soil and water organisms. So these are organisms that already exist naturally, even within the contaminated zone. Okay. So a lot of times all you have to do is add nutrient. So electron donor, which is a carbon source, and also electron acceptor, which could be oxygen, nitrate, sulfate, iron, manganese, or carbon dioxide. And just add those to the subsurface, and that would stimulate the microbes to grow. And organisms specific to the contaminants are selectively grown using the correct electron donors and the correct electron acceptors. So the correct carbon source, sometimes it's just as simple as adding something like vinegar, which could be a carbon source that would stimulate the native soil microorganisms. And if it's aerobic, you might add air or oxygen. If it's anaerobic, you might add nitrate or sulfate or one of the other terminal electron acceptors. Okay, and biostimulation takes advantage of natural selection. So once you have a contaminant in the soil, the presence of the contaminant will curtail growth to any bacteria that are going to be killed by it. So if the contaminant is toxic to the bacteria, then those bacteria will die. Any bacteria that survive, conversely, in the contaminant will remain. So these are resistant bacteria. And then some of the bacteria that survive will thrive and actually use the contaminant as a food source. Okay, and this just shows you some different zones. I don't necessarily like that slide, but you can see that when you're closer to the soil surface here, then and you get below the soil surface, you go from oxygen metabolism or aerobic metabolism to nitrate reduction to manganese to iron to sulfate, and then finally to methanogenesis, where the electron acceptor is carbon dioxide. Okay. Some of the different contaminants that can be cleaned up by bioremediation would be petroleum hydrocarbons. And you just want to inject, this says inject oxygen, but just inject air. You don't need pure oxygen. Just inject air. It has plenty of oxygen, and that will provide oxygen as the electron acceptor for aerobic microorganisms. The petroleum itself serves as the electron donor or carbon source. Chlorinated ethenes like perchloroethylene and trichloroethylene. This is a really slow process, but there are methanogenic microorganisms that are stimulated to eat or reduce the chlorinated ethenes, and the contaminants are slowly co-metabolized. So they're slowly eaten, usually in the presence of some type of carbon source or electron donor, and then the electron donor is just added until anaerobic fermentation takes place. Carbon tetrachloride, this is one I've worked on extensively, especially at the Hanford Nuclear Reservation. And it turned out that nitrate was a cheap source of electron acceptor. Denitrifying conditions were really, really good to get rid of carbon tetrachloride. And we used a cheap carbon source substrate, and that was added as an electron donor, and all we used was vinegar. Okay, you want to use something cheap because you're going to use a lot of it. So you don't want to use some type of fancy chemical compound. Even in some cases, sucrose or glucose might be too expensive. So in this case, we use vinegar. Heavy metal contaminants present a different problem. This would include things like chromium, lead, cadmium, mercury, cobalt, uranium, and others. And you can't ever get rid of a metal. You can't take a metal and make it a non-metal. It's an atom. And so it's conserved as matter. So what you have to do is you have to recognize that most oxidized forms of metal are water soluble, so they can actually contaminate a water supply. But most reduced forms of metal are insoluble and can be immobilized in the subsurface. So if there's a way that you can tie the metal to the soil and keep it from the water that people are going to ultimately drink, then that's the best strategy. So metals reduction is the best way that you can reduce contamination in water that might be used in municipalities or for farming or irrigation or whatever. So the strategy is to stimulate iron reducing organisms, and they will at the same time, that's concomitantly reduce and immobilize heavy metal contaminants. So you would just stimulate the iron reducers, and at the same time, they would just reduce the heavy metals. Once the heavy metals are reduced into their most reduced form, then they're no longer soluble in water, and they would not contaminate the water supply. And then you have a cheap carbon source that is injected into the subsurface to stimulate these bacteria. I've used cheese whey before, just the whey from the cheese making process made a great carbon source, and it's very, very inexpensive. And you don't have to add iron. Generally, there's enough iron in the subsurface to stimulate iron reduction. Here's a plot that just shows what the surface looks like in biostimulation. You have one well here that is injecting nutrient, okay, and it goes way, way below the soil surface. And then you have one well that is taking away the contaminant. It's called an extraction well. And then you have a series of wells that go down and monitor the progress of remediation. So here we have 5 monitoring wells. Okay, and you can see from the surface it's really boring, but if you look at the subsurface, then there is more and more activity going on. Here's a treatment zone where they use toluene and peroxide. Peroxide was to provide oxygen. Okay, and they had 2 bioactive zones. They had one here, and they had one here. This was below the soil surface, and this was in groundwater. It was where the groundwater or the water table started was here where it turned blue. And it turned out that there was an impermeable zone called an aquitard here that it was very, very difficult to pump nutrient through. So what they did was in one well, they remediated the groundwater above the aquitard. In the other well, they remediated the groundwater below the aquitard. Okay, so this was the bioactive zone is where the actual bioremediation is occurring. This is actual picture of a bioremediation site. It's really boring because the activity is going on in the subsurface. And here's actual data from a bioremediation site where they were taking perchloroethylene and selectively removing chlorines. Remember, perchloroethylene has 4 chlorines on it. You remove one, you get trichloroethylene. Then you get dichloroethylene. And then with a single chlorine, you have to get vinyl chloride. And finally, you get ethylene, which is easily remediated by aerobic organisms. So here's a site where they did remediation over a period of over 200 days. And you could see that the perchloroethylene levels went down very, very quickly. And then the trichloroethylene levels then came up and then went down. Dichloroethylene went up and came down. But the thing that was crucial here was vinyl chloride. Even though it's an intermediate of the contaminant perchloroethylene, vinyl chloride is more toxic than perchloroethylene. Vinyl chloride is known to cause cancer. And so we couldn't have vinyl chloride accumulate. And so as we were watching this remediation occurred, then we had to pay attention to this orange line where we barely got any, and that was good news. We barely got any vinyl chloride because if that came up as a product and started to accumulate, then we could create a greater hazard than what we had previously, what we had from the original contamination. Okay, here is biostimulation. This is uranium-reducing bacteria called Trionella onadensis, and you can see that it's got a lot of conjugation pili there, and it actually has precipitates of uranium on the bacteria itself. Okay, and I think for that, I'm going to switch gears here in the next slide. So I think this will conclude our first video lecture for Thursday, March 19th, and then there will be a second one that's coming up right afterwards.